Welcome to Finding Biomarkers that Cause GIST, from research to clinical application. During the seminar, all participants will be in a listen-only mode. There will be a question and answer session at the end of the presentations using questions submitted through the Q&A feature. Please remember the information provided in this web seminar is not intended as a substitute for your physician's guidance and care. My name is Sarah Rothschild, the VP of Program Services, and I would like to introduce you to our moderator for today. Dr. Margaret Von Maren is Chief of Division of Sarcoma Medical Oncology at Fox Chase Cancer Center. Her research interest focuses in developmental therapeutics for sarcomas and GIST. She has led investigator-initiated trials, collaborated with other investigators, sarcoma clinical trials groups, as well as industry-sponsored studies in the testing of novel agents in their rare diseases. Dr. Von Maren's work in sarcoma therapeutic development informs her role as the Associate Director of Clinical Research, in which she oversees the development of investigator-initiated clinical trials, the scientific review and prioritization process of trials, as well as conduct of clinical trials at Fox Chase Cancer Center. I'd like to hand this over to her as a moderator, and she can introduce the other speakers today.
Thanks, Sarah, and it's such a pleasure to be with you all today. It's my pleasure to introduce to you two outstanding young women who are doing research in GIST. First is Inga Marie Schaefer, who works as a pathologist at Brigham and Women's and Dana-Farber Cancer Institute, and also does research in GIST. And then second, we'll be hearing from Lori Rink, who I've had the pleasure of collaborating with for many years on GIST research. She is in the molecular therapeutics program at Fox Chase Cancer Center and runs a laboratory which 95% of the work that the lab does is focused on GIST, which is really exciting. So Inga is going to start first, and so let me turn it over to her.
Hi, welcome everyone. Thanks for joining this webinar today. It's my pleasure to participate in this panel discussion. As Max said, I'm primarily a pathologist, but I'm also doing a lot of sarcoma and GIST research in the Translational Research Lab. And it's my pleasure to show you some interesting leads from the lab today that you might find interesting and that we can also discuss about afterwards in the group discussion. So let me quickly try to share my screen. All right, so I'm gonna get started. So as most of you already know, GIST is really the poster child for targeted therapies. It was the first cancer for which targeted therapies have been developed, and that was a huge breakthrough in medical oncology. Most GIST are initiated by oncogenic KIT or PDGFR tyrosine kinase mutations, as you can see here on the right, showing these two tyrosine kinases. And as you also know, most GIST patients have traumatic and durable responses to tyrosine kinase inhibitor drugs, abbreviated as TKI in my talk, such as imatinib. For instance, you can see this patient shown on the bottom right who received imatinib and had stable disease even after 10 years on this drug. So this is really remarkable. However, we have also noticed that many GIST patients develop secondary tyrosine kinase resistance mutations during therapy. And this is quite challenging for the clinicians and also then for the pathologists to deal with. We observe that there is mutational heterogeneity in advanced GIST, which makes clinical management challenging. And what does that mean? On the left, you see a picture. This was a courtesy from my colleague, Dr. Chen Wout, who is a surgeon here at Brigham and Women's and the Dana-Farber, who was operating on a GIST patient who developed 46 metastases after treatment with imatinib and sunitinib. So there was really widespread disease in this patient. And we in the lab, in Jonathan Fletcher's lab, led by my colleague, Dr. Wang, sequenced these metastases. And you can appreciate just by looking at this picture that there was a huge range of different mutations in these metastases that conferred resistance to these tyrosine kinase inhibitor drugs. So that's really a huge challenge then if patients become resistant to imatinib and have different metastases, and all of them have a different pattern of resistance. So me working in the lab, I have identified an overarching goal for treating GIST beyond tyrosine kinase inhibition. And in doing so, the first goal is that I aim to discover new therapeutic approaches. And ideally, such new therapeutics would be effective in most patients with advanced GIST, and that would be irrespective of whether they have underlying KIT or PDGFRA tyrosine kinase mutations. And second, such drugs would be effective in most metastases within a given patient, irrespective of whether some metastases have become TKI resistant and what all these different mutations would be. So that's really the overarching concept of our research. And this would lead up to the discovery of new prognostic biomarkers and also predictive biomarkers that would tell us which patients would be most likely to respond to treatment. And I will show you a few insights now from my research. So let's go back a step to look at normal cells. So every cell in our body has to, divide and grow as tissue is growing and, for instance, healing as well. Other cells undergo death. So there's a constant turnover in the human body between growing and dying. And as the human cells grow, they undergo what is called the cell cycle. So they try to achieve and maintain the ideal size, growth, division, depth, and differentiation that is ideal for a specific tissue. And this is tightly controlled through a complex machinery of proteins, which is called the cell cycle, and which you can see on the right. The blue circles are cells dividing, and after division, the cell then undergoes again this cell cycle machinery, starting in the G1 phase, then S, G2, and M. M means mitosis, so the cells divide into two daughter cells. I'm not going to go into much detail here, but this is just to say that this is really an essential mechanism in every cell in the human body that is dividing. And this is tightly controlled because we don't want tissue to grow without control or too fast, right? So nature has put in place breaks or checkpoints in the cell cycle, which are indicated here on the right with these stop signs. So this is meant to prevent cells from growing too fast or when there's no need for growth. And also, you can imagine it to be like a road worker that says, stop, I have to repair, right? Because sometimes when cells divide, they accumulate mistakes in the DNA and they have to stop and repair these mistakes. Otherwise, it could cause cancer. So it's very important to have these checkpoints where the cell can check whether everything is going in the right direction. And if not, the cell would die so that you don't develop cancer. So this is what's happening usually in normal cells. But in cancer cells, the cell cycle is what we call broken or dysregulated. And this is actually a hallmark of cancer. Many cancers have dysregulation of the cell cycle because it's so crucial for growth. Cancer is a disease in which cells no longer respond to the social signals that normally govern their behavior. They can grow faster and without control. And they do that by removing these stops. So the cells can grow much faster than they usually would. And because these checkpoints are defective, also cells can now grow that have defects in their DNA, and that can cause cancer. And our work currently is directed at finding ways to really restore these breaks on the cell cycle in cancers that have lost these breaks. This is a schematic. It looks very complicated, so I'm not going to go into much detail, but this is just to show you this gray circle in the middle is again the cell cycle, and now we have shown in purple and blue the different proteins and regulators that govern this machinery. And it's a complex system that I'm not going to go into much detail here, but our work really is focusing on these proteins and how we can target them in sarcoma and GIST. And I will show you now why cell cycle regulators, meaning these proteins, are promising targets for cancer treatment. We are mostly focusing on this part here that I've shown on the right, which includes the P16 tumor suppressor, which is usually inhibiting growth. So it inhibits cyclin D1 and CDK4/6, who work through RB and then let the cell cycle progress. So I've shown a couple of years ago in my postdoc work, when I was still a postdoc, that one of the first tumor suppressors, meaning those proteins that stop growth, that is defective in GIST, is called MAX. It is located on chromosome 14q, and I have shown that MAX stimulates P16, which is good because P16 is also one of these break proteins that stops the cells from growing, and it's inactivated in up to 70% of GIST. So that shows that MAX really is, and it occurs already in micro GIST and very early GIST. So MAX is an early tumor suppressor that's inactivated in GIST. And this is the first hint for why we think the cell cycle is really a great target in GIST, because we know that it's important, this whole machinery, in at least 70%, if not all of these tumors. I will just quickly show you here. So this is a low-risk GIST that has no aberration in MAX. Shown on the left, you can see these brown dots are the cells that express this protein. And on the right, you can see that P16, this tumor suppressor that's regulated by MAX, is intact, meaning it's also brown in the tissue, so it's expressed. And then we had the opportunity to eliminate MAX. Here on the left, we have a GIST that does not have MAX expression. You can see the cells are blue, so the protein is missing. And then you can see here on the right, in this tumor, P16 is also gone. So this shows how directly these two proteins are interconnected. And then if we look further beyond MAX, we can actually say that GIST are progressing through a sequential series of genetic events that incrementally dysregulate the cell cycle. So on the left, we have the more benign or early GIST, and then on the right, over a few steps, they develop into becoming malignant and aggressive GIST. And we're really looking into why this is the case and what are the different steps that make these tumors more aggressive. And this was also some work with our colleague, Mike Heinrich, and the Fletcher lab that shows, if you look here, shown at the bottom, that any event, meaning any cell cycle regulator hit, occurs in only 70% of low-risk GIST, but in almost all high-risk GIST and virtually all metastatic GIST. So this again shows us that this is a really crucial pathway in these tumors. So to go back to my first goal, I think that the cell cycle really is a crucial mechanism in GIST, and that a drug that would target the cell cycle would indeed be effective in most GIST, most patients with advanced GIST. And I've also shown that this is irrespective of whether they have an underlying KIT or PDGFA mutation. So this is already leading us in the right direction. And now I want to briefly step back from GIST and look at breast cancer. So breast cancer is, as you know, a very common cancer type. And in these tumors, people also found that the cell cycle is commonly dysregulated. And we again look at the schematic. And for breast cancer, recently, 3 drugs have been FDA approved that can inhibit this crucial player, CDK4/6. And in breast cancer, so these are these drugs. They are called ribociclib, palbociclib, and aberrations. And they have really been very, very promising in breast cancer. And people have also found that if you combine them with aromatase inhibitors, meaning hormone-directed therapies, they can even be more effective in these cancers. So this is a good model for us working in GIST and sarcoma, that we know of a cancer type that responds to these drugs very well. And this is from a different paper where this CDK4/6 inhibitor drug called Palbociclib was tested. On the left, you can see a patient with ovarian cancer that responded very well to this inhibition. The right line indicates the tumor suppressor marker CA125 in blood. And you can see after Palbociclib was added, after 2.5 months, this tumor suppressor goes down rapidly. And on the right, this was actually a GIST patient that was shown in this publication, a 68-year-old male with metastatic GIST who had inactivation of this tumor suppressor CDKN2A P16 that I've shown you in the schematic. And this patient also responded to Palbociclib single agent treatment with partial response lasting 11.3 months. You can see that at baseline and after 2 months, these metastases that the patient had in his liver and in his chest became much smaller. So this is already good leads that this drug can be effective in GIST as well. And to quickly go back to this question of heterogeneity, meaning that we have so many different metastases in patients with metastatic GIST that can be challenging to treat. Shown on the left is this patient that I've actually shown you at the beginning, where we had that picture from the surgeon with many, many different metastases, and all of them had different resistance mutations. But we found that the same patient had mutation of this cell cycle regulator MAX, and the same mutation in all of these metastases, and also TP53 mutations can be conserved across all metastases. So this really indicates that if we target the cell cycle, we can hope that this would be effective in most metastases in a given patient, irrespective of whether some of these have become TKI resistant. And to briefly now touch on biomarkers, because I know you are all interested in the development and what we find or can discover as new biomarkers in GIST. This paper has shown that the presence of cell cycle dysregulations is associated with a poor prognosis in GIST patients. On the left, you can see in blue, the line at the top, these were patients with GIST who did not have cell cycle hits. And you can see that after 125 months, few patients died, but 90% survived. However, the patients who had cell cycle hits, at least one of them, they died much faster, indicated by the red arrow. And this was progression-free survival, but the same is true for overall survival, shown here on the right, where the patients without cell cycle hits did so much better than those who had cell cycle aberrations. And this could lead into, in the future, establishing biomarkers that indicate how many cell cycle hits a patient has. And that would indicate that this particular GIST is associated with a poor prognosis and higher risk. And this is just an example of how we could in the future maybe apply these biomarkers. So I'm just showing you immunohistochemistry pictures. On the left, we have P16 staining, in the middle RB1, which is a key regulator in the cell cycle, and on the right, TP53. So these are all tumor suppressors. They are expressed in this normal tissue at baseline weekly. RB is a little bit stronger. And then let's look at a metastatic GIST. So we can see at the bottom that P16 is very strongly expressed in this case, and that is because RB1 downstream is lost. So this important tumor suppressor is lost in this patient, leading to upregulation of this upstream protein as a compensation mechanism. And also TP53 expression was abnormal. So you never see such strong staining usually. That means there's also a mutation. And this is just to show you that we have the tools at hand and we are refining them on different levels to study these proteins, and that what one could imagine to use these in the future as well. To conclude my talk, I have shown you today that cell cycle dysregulation is really a hallmark of cancer, and we are trying to leverage this as a vulnerability in GIST and other sarcoma. Cell cycle restoring agents can restore the breaks in the cell cycle and turn the cell back to having more controlled growth that is not independent. And they are effective in other cell cycle altered cancers, as has been shown very impressively for patients with breast cancer. And our studies show that most advanced GIST have cell cycle regulator aberrations that can be a rationale for evaluating therapeutic cell cycle restoration. And I hope to show you some more about this as my work evolves over the next months and years. And I look forward to answering your questions and to discussing with you. And I would like to thank all my colleagues and friends at Brigham and Women's Hospital and Harvard Medical School. Thank you very much.
Thanks, Inga. That was really informative. I think what we'll do is move on to Lori's presentation, and then we can end with questions and answers all together. So Lori.
Thanks, Meg. I'd like to say good afternoon and thank you all for coming. And thank you to Sarah and the LifeRack group for the invitation to discuss some of our recent translational efforts in GIST. So today I'll be talking a little bit about our attempts at identifying new targets in GIST using a method called kinome profiling, which I'll be describing in a bit. So where are we at in terms of therapeutics for GIST? There are currently 5 therapies approved for GIST treatment: imatinib, as Inga just discussed, sunitinib, regorafenib, avapritinib, and ripretinib. And the last 2 have been just approved within the last year, so making great progress. We know that 75 to 80% of all GIST will respond to imatinib upfront, but that still leaves 20% of the GIST population that doesn't respond to Gleevec. And we know this is because not all GISTs are created equal. It's really important to know the specific mutation that's in a given tumor, because it's been shown that the genotype or the mutation in a tumor can correlate to treatment response. In addition, in the case of acquired resistance, when a tumor becomes resistant to imatinib, knowing the secondary acquired mutation is also important in informing treatment decisions. So again, a lot has been learned in the last 10 to 15 years in studying GIST, and we now appreciate the fact that there are 3 major molecular subtypes that include KIT mutant, PDGFR alpha mutant, and succinate dehydrogenase deficient, or SDH deficient tumors, which were previously known as wild type tumors. And although all of these GIST share smooth muscle and neural components, as well as staying positive for KIT and or DOG1, we appreciate the fact that there are significant differences in terms of clinical histories, different genomic profiles among the subtypes, and different profiles in terms of response to imatinib and other therapies. So for instance, KIT mutant tumors tend to be more aggressive and grow faster compared to PDGFR alpha and the SDH deficient tumors. Interestingly, if you look at the tumors and the morphology of the cells that make up the tumors, KIT mutant tumors have spindle-shaped cells, kind of round in the middle and tapered at the ends, whereas SDH deficient tumors are more like circles, round epithelioid-shaped cells. And PDGFR alpha has a kind of mixture in terms of the morphology. In terms of treatments for these subtypes, KIT mutant tumors tend to be the best imatinib responders. Although acquired resistance is a clinical problem, sunitinib, regorafenib, and ripretinib are approved for 2nd, 3rd, and 4th line therapies. In terms of PDGFR alpha tumors, the majority of these tumors have a mutation called D842V, which are very resistant to imatinib. And fortunately, in the last year, a new drug, avapritinib, which is a selective KIT and PDGFR alpha inhibitor, has been approved as the frontline therapy in that subtype of patients. Although already clinically acquired resistance has been observed in some patients to this drug, and this drug has also been associated with some cognitive side effects, especially at higher doses. In terms of the SDH deficient GIST, they are often more resistant to imatinib. They may respond a little bit better to sunitinib, but unfortunately there are no subtype specific approved therapeutic targets. So in recent study from our group, we sought to kinome profile GIST tumors from these different subtypes in order to potentially identify new targets that we can go after. Kinome profiling, as I'll talk about, involves really looking broadly at a class of proteins called kinases. And so you may say, well, why study kinases? Protein kinases are key regulators of cell function. They make up one of the largest and most functionally diverse gene families. So there are 518 protein kinases in the human kinome. These proteins are really important in directing activity and localization and functions of proteins inside of the cell, and they really kind of orchestrate the overall activity and many cellular processes inside of the cell, including the cell cycle, which Inga so nicely described previously. We know that 85% of the human kinome is involved in diseases, including cancer. But we also know that this is kind of an understudied field in that only 80 kinases have been targeted thus far. We also wanted to study kinases because these are a highly druggable class of proteins. There are currently now 60 different FDA approved tyrosine kinase, or I shouldn't say just tyrosine kinase, but kinase inhibitors. And many of these are used to treat different types of malignancies. The other reason kinases are so important is that we know that there are more than 90 different kinases that are what we call drivers and drive tumors. And two of these you're very familiar with, KIT and PDGFR alpha. These are kinases that are drivers. Typically, a GIST is formed when there's a mutation called a gain of function mutation in either KIT or PDGFR alpha. And when that mutation occurs, it kind of flips the switch and turns that kinase on all the time, kind of unregulated. And when those kinases are on, they're activating all these downstream signaling pathways inside of the cell that lead to increased cell proliferation or cell growth, inhibition of cell death, and ultimately lead to tumor formation. And over the years, the GIST field has really studied extensively these pathways, but we believe that these pathways, as it's described here, is kind of oversimplified. And what may be the case is really something more complex with many more kinases involved in GIST biology. And so we sought to really look at a greater fraction of the human kinome to see if we can learn some insights about GIST biology, but also to identify new targets. And so what did we do? So we performed this kinome profiling experiment in collaboration with James Duncan at Fox Chase Cancer Center. And what we did was we identified 33 GIST tumor samples. These were samples that patients consented to give for research purposes. They were all untreated, and they were from the 3 major subtypes of GIST. And in addition to those 33 tumors, we also profiled 9 normal gastric tissues. And what we did was we basically isolated protein from each of these tumors, and we run them on a column that has beads, and those beads bind to the kinases. We elute those kinases out, and we put them onto a machine called a mass spectrometer. And the job of this machine is to identify kinases and also to quantify the abundance of those kinases in particular tumors. And the goal was to identify new targets in each of those subtypes. So what did we find? Well, we found that we could measure 296 kinases. This represented 60% of the human kinome, so it was pretty good effort here. And when we looked at how closely the samples from each of the subtypes kind of clustered together, this is called a PCA plot, or a principal component analysis. We first noticed that all of the GIST, which are these red, blue, and green dots here, clustered together, kind of distinct and separate from the normal gastric, which is in pink. This is not surprising, right? Because these have the gain of function mutation. They're activating all those downstream signaling pathways. And so you would expect them to be very different than the normal GIST. But when we looked at the subtypes, we were interested to find that here's the wild type or the SDH deficient GIST, which are clustering together, those green dots. Here's the red dots, which are the PDGFR alpha mutant GIST, kind of tightly packed together, indicating that there were differences in the kinome between the 3 subtypes. The blue dots here represent the KIT mutant tumors, which were kind of in between the SDH deficient and the PDGFR. So we're currently right now working on validating and functionally characterizing those kinases that we found that were unique in the different subtypes. And I hope to be able to report more on this in the upcoming months. We next decided to take a different approach where we looked for kinases that may be activated and overexpressed in all mutant GIST, so in both the PDGFR and the KIT mutant subtypes, compared to normal gastric tissue. And again, we found many kinases that were up in the mutant GIST compared to normal. Many of these kinases, like protein kinase C theta, FGFR1, KIT, PDGFR alpha, these were all kinases that we've long known in the GIST field to be important in terms of GIST biology. We also interestingly found several kinases that were cell cycle related kinases like WEE1 and CDK4. And then completely understudied kinases also were identified, and we're still in the process of really working through the data, validating many of these and functionally characterizing them. One way that we chose to functionally characterize and to kind of prioritize which kinases we were going to go after first was to perform a functional study where we received GIST cell lines from Mike Heinrich and Lily Klug at OHSU. And these cell lines, one of them is a KIT-driven cell line, and one of them is a PDGFR cell line with that D842V mutation. And what we did was we took each of the kinases and we knocked down expression of the identified kinases in these cell lines to see what effect the knockdown of those kinases would have on cell viability, on how many cells died when you knock down those kinases. And so the big bars here that say control, these are negative control. This is when we knock down a gene that has no effect on viability. And you can see 100% of the cells are still alive. In comparison to our positive controls, so when we knock down KIT in the KIT-driven cell line, or we knock down PDGFR in the PDGFR-driven line, you can see a high level of cell death and just a small fraction of the cells that are still alive. When we looked at some of the knockdown of the identified kinases, we were interested to see that one of those cell cycle kinases, WEE1, which is a G2M kinase, related kinase, when we knocked down that kinase, we were able to see a lot of cell death and minimal viability, almost comparable to when you knock down KIT or PDGFR in these cell lines. So this indicates that perhaps this kinase plays an important or critical functional role for GIST cell survival. So from there, we wanted to check the feasibility of targeting this inhibitor or this kinase in GIST cells. And so we were fortunate that there's already a WEE1 inhibitor, a selective inhibitor available. This inhibitor is in clinical trials being investigated in other tumor types. And we looked at this inhibitor as a single agent and in combination with avapritinib. And we did this using a spheroid assay. A spheroid assay is when we take those same cells I just mentioned before, and we grow them in small tumors or spheroids. And these cells form tight cell-to-cell contact. They're grown in tissue culture dishes and incubators, but they look like mini tumors. And this is, I think, a better assay than growing cells monolayer because they more closely mimic the physiology of tumors. So we can look at what the spheroid looks like when it's treated with no active drug, nice tight kind of circle of dense cells. When we treat with the WEE1 inhibitor, which is this second row here, what you can see is that the spheroid is starting to break apart and you get this halo, these dead cells that are kind of breaking off of the spheroid and dying. Here's avapritinib as a single agent. So you can see that they're still kind of tight spheroids, but they're much smaller than the untreated spheroids. And then the combination, you've dramatically decreased the spheroid. You have just a very small volume with lots of dead cells surrounding that spheroid. So when we do these studies, we do lots of repeats and many, many experiments to confirm these results. So just below here, I'm showing you kind of the quantification of and the summary of many experiments where you can see that when we look at the spheroid volume, combination has far smaller volume compared to either of the single drugs. The other thing we can do is we can break open those spheroids at the end of the drug study, and we can look to see how metabolically active, how viable the cells are within those spheroids. And you can see here that the combination looked much more effective than either single agent. So because of these strong spheroid data, we next moved into performing GIST xenograft models. And a xenograft model is when we take those cell lines and we inject them into immunocompromised mice, and we grow small GIST tumors in mice and treat them with various drugs. So in this study, we did no drug, avapritinib alone, WEE1 inhibitor, or the combination. The combination is in green. And we have two kind of readouts for these xenograft studies. The first is tumor volume. So we're measuring the tumor volume in these mice over time. And what you can see here is that in green, the combination allowed for a significant tumor stabilization in these mice. In the KIT mutant, you can see that the avapritinib worked pretty well, but then eventually it's starting to grow, whereas the combination remained flat. And we had a significant stabilization with even some tumor regression, significant shrinkages here in the PDGFR mutant model. Down below is a readout of overall survival of the mice. So you can see that the green lines indicate that the combination is improving the overall survival of these mice, most significantly in the PDGFR-driven tumor, where at the end of the study, which was 89 days, and 89 days is a lot of days in mouse studies, 75% of the mice were still alive at the end of the study. No other mice in any of the treatment groups were alive at that point. So significant, it seems, synergy between these two inhibitors. So the final thing we did was, and we always do this, is we look to make sure the drugs we're using are hitting their specific target. And we do this using an immunoblot or a Western blot, where we treat the cells in culture with the drugs, we harvest protein, and we use specific antibodies to detect whether the drugs are hitting their target, which they did. But one of the things we noticed is that in the cells treated with the WEE1 inhibitor, or in the combination, we saw increased or elevated levels of DNA damage and increased and elevated levels of cell death. So we are hypothesizing here that this increased DNA damage may be a result of the loss of cell cycle checkpoints and decreased time for DNA repair mechanisms, ultimately causing increased cell death. And so just to conclude, we feel kinome profiling is a strong method to identify potential new targets in GIST. We showed that the kinome profiles among the different GIST subtypes are different. And we also showed some similarities in the case of WEE1, where WEE1, which was identified from the kinome profiling, could be inhibited in combination with avapritinib to increase efficacy in both KIT-driven and PDGFR-driven cell lines and animal models, and perhaps provide strong preclinical justification for its use in GIST. And so I'd like to thank you all for your attention, and thank the Fox Chase GIST group, as well as some of our collaborators at Fox Chase and some of the outside collaborators that we have.
That was great, Lori. Thank you so much. So we've had some questions come in, and so I'm going to try and maybe put them in some order to sort of make the conversation flow. So I think the one question for you, Inga, you talked a lot about the cell cycle and its role in GIST, and also talked about GIST with mutations. And the question came up is, does this apply to tumors that don't have mutations, say the SDH deficient tumors that Lori mentioned, or maybe some of the tumors that in the past we qualified as wild type, but we understand, for example, are associated with NF1 mutations or other classes of much rarer subtypes of GIST? You're on mute, Inga.
That's a great question. Thank you. Yeah, as I've said, and also as Laurie has shown, for us, it's important to find a drug that's ideally applicable to all patients with GIST, right? Whether they have KIT, PDGFA mutations, SDH mutations, NF1 mutations, or what has been called so-called wild type GIST. We often find mutations in the SDH genes, though, in these so-called wild type GIST. So I think a very, very small subset of these are truly wild type for any of those four key players. So to answer your question, I think we have promising results. They have not, we are still working on it, so I cannot provide much more information, but there's evidence that yes, the cell cycle is important, whether it's in KIT mutant GIST, SDH deficient GIST, or an NF1 mutant GIST. And we also have early leads that the drugs that we are testing, and whether a single agent or combination, might also be effective in SDH deficient GIST.
That's great. sort of speaking about the mutations, somebody asked whether or not there was a difference between mutations that are caused by deletions or by insertions. Does that affect what we see in terms of the biology of the tumor, or they're equivalent?
Yeah, I think the attendee who asked that question was probably referring to the mutations in KIT, right? So yes, we know that there are subtle differences in terms of whether you have a deletion or a point mutation in KIT exon 11. So the deletions usually have a slightly more unfavorable clinical prognosis versus the point mutations. Usually deletions and insertions are I think, biologically, it's often the same thing. It's kind of, some genomic reports call it an indel, or others just call it a deletion and an insertion. So this, I would think, can be viewed as the same. It has the same biologic implications as opposed to point mutations in KIT exon 11. However, this is a very, very subtle difference. We know that KIT exon 11 mutations in general respond well, if it's a primary mutation, to the standard dose of imatinib, and that KIT exon 9 mutations have slightly less sensitivity towards imatinib. But within exon 11, they are quite similar, I would say. Meg, you are the medical oncologist, and you can maybe attest to that. But I would say within exon 11, the differences are subtle. And this is, so we're just talking about primary mutations. That are the mutations that are there, that cause the GIST, and that are there before you start treatment.
Yeah. So yeah, there occasionally have been some that have been described that maybe have a little bit more concerning in terms of prognosis, but overall, I think that what you said is very true. And then the last specific question for you, Inga, is how do we test for cell cycle events? You showed some work that you've done in terms of immunohistochemistry, like taking the tissue and studying it in the pathology lab, but are there ways of doing that, say through blood tests or other ways that we could look at that?
Yes, so we can do that. In theory, it's possible to do that, as you can test for KIT or PDGFA mutations either before treatment or during treatment to see if you develop resistant clones, right? You could use the same technology to test in the blood for any tumor DNA or tumor cells floating around in the blood that could have mutations in these cell cycle genes. And the most important genes would be RB1, CDKN2A, 2B, TP53. So these are the most common tumor suppressor genes. So most panels, would be able to incorporate these genes if they are not already in there. So yeah, the answer is yes. Ideally, you would, however, as a pathologist, first take out the primary GIST and do the examinations, test the tissue for mutations, and then later on during treatment, you can then kind of monitor how well patients respond by doing the blood test, because it's less invasive. You don't always have to do biopsies, and you can earlier and easier detect possible recurrence when the tumor comes back. But ideally, you would first start with the tissue and then go to the blood test afterwards.
Great. Lori, you talked a lot about looking at different subtypes of GIST. And from the work that you've done, where do you see treatment going in the future? Is that going to become more and more tailored?
So I think there are definitely, from the kinome profiling, you see distinct differences. Other groups have shown genomic profiles are very different. For instance, Ron DiMatteo's group showed that in the PDGFR mutated tumors, there seems to be more immune cells infiltrating into the tumors. So I kind of envision maybe different groups going in different directions in terms of finding novel subtype specific findings that there are therapies against, maybe somehow using the immune system to target PDGFR tumors compared to KIT. But I think the approach that Inga described and the WEE1 example that I gave of trying to find something that could be used irrespective of the subtype could be beneficial for patients as well.
Yeah. Has there been any research on looking at kinases involved in NF1 associated GIST that you're aware of? And that Lori or Inga?
Yeah, so we didn't profile those, and I don't know of any literature looking at other kinases in NF1. I don't know, Inga, do you have any ideas?
I can just quickly mention that when we studied MAX, the tumor suppressor that's on 14q, that's an early hit in GIST, that is the second hit after the KIT or PDGFA mutation, that we found that these events occur in NF1 mutant GIST as much as they do in KIT and PDGFA mutant GIST. So we think in this regard, the NF1 mutant GIST are comparable to the regular tyrosine kinase mutant GIST in terms of cell cycle alterations. But for us in the lab, for us researchers, there's always a challenge. So we have a concept in mind, we want to test it. So we usually start using GIST cell lines to see if our hypothesis can be tested, right? And for NF1 mutant GIST, there's a limit of models that we have available at hand. So we usually test these hypotheses first in KIT in KIT mutant cell lines. There's also very few PDGFA mutant cell lines, I have to say. So that's the only reason. But I think in terms of cell cycle alterations, NF1 GIST are possibly comparable to KIT and PDGFA mutant GIST.
So I know from the clinical perspective, when you look at sort of NF1 and the pathways that it's involved, it does seem to involve MEK as one of its other kinases is involved. And there are now some MEK inhibitors. And there have been occasional reports suggesting that MEK might be a way of targeting those NF1 type tumors. I think the other thing to say about those tumors that oftentimes, while some of these patients may have many tumors, they oftentimes grow very slowly and are more indolent, which is a good thing for the patient who has them. But there are some tumors that are associated with NF1 that are not necessarily malignant, that have recently been shown to respond to MEK inhibitors. So I think that's something that could potentially be tested, and hard to get a full clinical trial potentially just because of the rarity of those types of tumors. So from the work that you did, Lori, there was a question about whether or not the combination of the WEE1 inhibitor and avapritinib, would that increase toxicity? And maybe you can speak to your experience in animals and what you saw there.
Yeah, so I think one of the benefits of thinking about a combination approach is that by hitting two different targets, rational targets, that you could use lower doses of both drugs and maybe decrease some of those toxicities. This could be really important in terms of the avapritinib. As I mentioned in my talk, one of the side effects that are becoming known is that at higher doses, there's a lot of cognitive toxicities, which were not seen with many of the other TKIs. And so that's concerning. And so the thinking is, if you could decrease significantly the dose you give of avapritinib in combination with WEE1 still at a lower dose, perhaps it could actually not amplify the toxicities, but decrease the toxicities. And I think there have been studies in patients looking at WEE1 inhibitors as single agents, and they're relatively well tolerated. So I think there's hope for maybe inhibiting some of those toxicities.
Yeah, and one of the questions that's sort of come up throughout the chat and as we're talking about these different agents is, when will we see them in clinic? So I think I'll address that just because as part of what I do, as clinical researchers who are looking for new therapies, we are dependent on researchers like Inga and Lori to provide us with preclinical data that suggests that it works. The next step is really to be able to convince funding agencies or pharmaceutical sponsors that testing it in patients makes sense and that there's a potential benefit. And once that happens, there are stages of clinical research that we do. There's the first stage, which is phase 1, which is looking primarily at what's the best dose and what are the side effects. And I think increasingly phase 1 studies, which in the past we sort of let anybody join who needed some form of therapy. But with these targeted therapies, more and more phase 1 studies are really becoming very disease specific or for a subset of diseases. And therefore, patients are much more likely to benefit from participating in phase 1 studies. Once you sort of figured out what the toxicities are and what the best dose is, then you can really evaluate the efficacy and do a phase 2 or greater phase 3 study, which will then lead to potentially to approval. Sometimes, particularly in this country, if a agent is approved for another indication, we can sometimes use it even though it doesn't specifically say it's approved for GIST. That is, for most physicians, we want to make sure that there is information on safety and also on efficacy from prior clinical research. So that's a 2-minute sketch on how do we get there. Certainly, I don't know where Inga is in her work and the collaborators she has up in Boston at Dana-Farber that have a very active GIST group, but certainly in my sort of the plans of in the next 6 months, trying to research and identify, can we get a study funded, supported, approved, or drugs able to be used from the ones that have been tested clinically to date to test the concept that Lori has tested. Inga, I don't know if you have any information about where you are in clinical testing.
Yeah, so I'm still in the laboratory, testing what we call in vitro and in vivo. So in the cell lines, the GIST models that we grow in the dish, and also in mice. I have some promising approaches, but these need to be, I need to wait what the results will show. But I hope that, within maybe the next 1 to 3 years, this might hopefully lead into a clinical trial. But yeah, I'm on the right way, but it takes a few more months, I think, at least to say for sure.
Great. We've been talking a lot about testing tumors, and somebody asked the question about genetic testing, and I think that this is something that's really important to sort of clarify. Me as the doctor who sees patients, I think about genetic testing of the tumor, which is really important to help guide with therapy. And somebody said, if your doctor is not doing it, is that malpractice? Well, I would say in this country, we highly recommend, and I think now, especially that we have drugs like avapritinib that are so tied to specific mutations in terms of efficacy where there is no other drug, that it is very important to get mutational testing. And I think really every person who's considering getting some form of therapy needs to have their tumor tested. But then there's the other question about genetic testing for, is my tumor that I have, can I somehow share that or pass that on to my children or my siblings? And that's what a distinct type of testing, which is germline testing, which is really testing changes in the DNA of all the cells of your body. And for men, that includes your sperm. And for women, your oocytes in your ovaries, which if you have children, can be passed on to them. And so there are certain types of GIST where that is more important to do. Certainly the SDH deficient tumors are one. NF1 associated tumors are another, because that is can be a germline, and not always, but it can be a germline associated tumor. And there are both of those are tumor types or genetic syndromes that are associated with other cancers. So not only for other GIST, but other types of cancers. And there are also some families, not common, that have inherited germline mutations in the KIT gene or the PDFR alpha genes, where you may see multiple types of GIST. Some of them have also had some associated melanomas and other tumor types. So I think it's an important question to ask your physician about if there's any concern for testing of your tumor, which is very important, but then also you yourself and whether or not you should be seeing a genetic counselor and asking about genetic testing. All right, I'm scrolling quickly because we've had lots of questions, which are great. One that I'll add, because it's sort of associated, TP53 or the P53 mutations that we talked about. Does that important to identify because treating it can help prevent recurrence, or is it just seen, are there therapeutic options that you're aware of?
Yeah, I could maybe answer that question. So there are currently no therapeutic options that would directly target TP53, but there are many labs that I know of that are working on that. And TP53 in itself does not, predict recurrence. It usually occurs in the GIST that are at higher risk or metastatic. It's usually not present in the low risk GIST, but a TP53 on its mutation on its own does not tell you whether the GIST is going to recur or not.
Yeah. So I know that there currently, there's one study that's looking at immune approach for specific types of P53 mutations. It's a study at our center. I think people have for a long time wanted to target it just because it is so important in cell growth, cell regulation. And so it is definitely a target that we think is viable potentially for helping control malignancies, including GIST. So I'm gonna, we have about 3 minutes left. There have been a couple of questions about funding for GIST research. Maybe Lori, you might wanna talk about where you've gotten funding, and Inga as well, and how you've been able to do the research and your thoughts about that.
Sure. So I've been fortunate to have a variety of funding sources. I've been funded by the National Cancer Institute. I think it's challenging studying a rare tumor type to get some of that federal funding, those dollars, because a lot of the times they go to more common tumors. But I always kind of stress in my grants that studying the biology of these rare tumor types can not only provide insights for patients with GIST, but also for other tumor types. And so I think it's important to make sure people know how important studying rare tumor types is. We've also, Meg and I have been blessed with a lot of philanthropy and different foundations who have acknowledged how difficult it is to receive the federal funding for studying rare tumor types. And some of that funding from these foundations have been really helpful in terms of generating pilot data and asking questions we couldn't normally ask before writing the larger federal funding grants.
Yeah, and I can just echo what Laurie has said. It can be quite challenging for someone who really focuses all their efforts on GIST or these rare sarcoma tumors to get federal funding, but I've also been lucky to receive funding from the NIH National Cancer Institute. And my research is really enabled through that. And also I was fortunate to receive funding from a foundation, which is called SARC. So yeah, these two grants currently support my work. Yeah, and I can just say echo what Laurie said. It's important to explain to those people in charge that we think GIST has been a poster child for the development of targeted therapies, and we believe that it can continue to have that role for other crucial therapeutic approaches that can be then translated to other cancer types.
Great, thank you. And Sarah's back.
I am. I just wanted to say thank you. This was an incredible conversation. It's so exciting to see women in medicine that are advancing research in this field. I know that there was a lot of excitement among our community about the presentations that you gave today, and I'm sure they want to hear more and they want to hear updates. So I hope the LifeRaf group can continue to be that communication vehicle to our patient community to hear those updates and see how we can translate that onto the patient community, especially with what you're doing in the lab. And as it gets translated into the clinic, helping them be aware of trials that are available and new research. And of course, the LifeRaf group is also really excited to make this information available to the community. We used to support research, and we hope to continue to do so in the future. I think all of you are doing great work, and I hope that the LifeRaf group can continue to be a part of advancing the field through research opportunities. I just want to conclude and also let everybody know that this presentation is going to be, it is recorded. It will be available next week through our YouTube channel. And we are launching this testing campaign. So I think everybody who is today a part of this live event or who will be watching the recording, I think this is part of arming yourself with more information about how important it is to get testing. I think you should hear Meg's words very loud and clear, how important it is to get testing. Everybody, all the GIST patients, it's so important. So please speak to your provider about this. And if you have questions about how to ask your provider about it, feel free to contact the LifeRaf group and we can assist you with that. So I want to thank everybody today for joining us, and have a lovely day. Thank you.
Thank you.
Thank you.